Hello and welcome. In this video, I will be demonstrating and explaining the process of meiosis. First, we have stage 1 of meiosis, interphase. During interphase, both the mom and dad chromosomes are bundled up into the nuclear membrane and are not visible. The next stage is prophase 1. During prophase 1, the nuclear membrane dissolves and the chromosomes split up. You will see that there are 4 chromosomes, 2 large and 2 small. The blue chromosomes are the dad's chromosomes, and they have informations of the dad's DNA. The orange chromosomes are the mom's DNA, and the large chromosomes can have information like something like eye color, and the small chromosomes can have information about something like hair color. During prophase 1, the mom and dad chromosomes do something called crossing over. So basically, they will go on top of each other like this, and they will actually share DNA, and it will look something like this. I know there's a discrepancy in the color, but it would look something like this. Usually it would be the same color as the chromosome, but it would almost wrap around like this, and that would be the mom's DNA is now on that father chromosome, and vice versa. The father's DNA would be around the mother's chromosome. This is called crossing over. At this point, these would be called diploid cells. The next stage is metaphase 1. During metaphase 1, the homologous chromosomes line up across from each other at the equator. Every time meiosis happens, these chromosomes can line up in different formations. For example, one time they can line up like this, or another time they can line up like this. Both dad chromosomes are on one side and both mom chromosomes are on one side. This is called random assortment. During metaphase 1, the spindle fibers will also appear. And it would look like this, like they're going to attach the chromosomes on each side. The next stage is anaphase 1. During anaphase 1, the spindle fibers attach to the chromosomes and they will pull each chromosome away from the equator out to the pole of the cell. Just like that. And now the cells would be separated from the equator and on each side. Next up is telophase 1. During telophase 1, the chromosomes will be pulled to the poles and cell furrowing will appear. Cell furrowing is when the cell almost appears to be getting pinched, sort of like this, so it goes in like that, gets pinched in. And this is preparing for the cells to actually split and reproduce into 2 different cells. So next is cytokinesis, which is basically the process of the cell splitting into 2 different cells. So it will split up, and basically what will happen is it'll be a brand new cell, and there will actually be 2 of them. And each cell will have one father, the chromosome 1, and one mother, chromosome 2. After cytokinesis, prophase 2 begins again, but this time there is only one copy of each chromosome present, and the nuclear membrane briefly reappears before dissolving again. Next is metaphase 2, where the 2 chromosomes will line up at the equator of the cell again, except they'll line up in a different formation, and instead of being like this, lined up like this, across from each other, they'll actually line up like this in a single file line. And the spindle fibers will also reappear and will look something like this. Okay. And after metaphase 2, anaphase 2 will happen. But differently from anaphase 1, the spindle fibers will not pull the chromosomes away from each other, but they will pull the sister chromatids and pull one of each chromosome sister chromatid to the poles of the cell. And it will look like this. And it would look just like that. So each one of them will have one sister chromatid at the pole of the cell again. And next will be telophase 2, where the cell furrows reappear again, and they cut inward like this, and they start pinching the cell again, until eventually it'll split into another 2 cells. So now there will be 4 separate cells, each will be like this, it'll look like this. Each will have one one sister chromatid from the dad's DNA and one small mother chromatid. And they will also have the nuclear membrane around them again. And there'll be 4 of these. So there'll be 2 more of those, one with a big mother sister chromatid and a small father chromatid. And yeah, that's about it. These would be called haploid cells at this point, and that would be about it. That explains meiosis. Thank you for watching.